Okay, in the last video I introduced the idea of protein synthesis, and we talked about the different structures that are involved. So now we're going to talk about the two main steps that are that make up protein synthesis. Okay, we'll start with start with transcription, which is going to be step one. Okay, the DNA code, the series of A's and T's and G's and C's in different sequences that make up the genes, are copied from the DNA onto a strand of messenger RNA. Okay, and we often say that the DNA, since it's double strand, is zipped open, and the mRNA is made based on one side of the DNA. Now, that's called transcription. In step two, the mRNA is going to travel outside the nucleus, so it's taken the instructions from the DNA. It's going to travel outside the nucleus into the cytoplasm, where it's going to find and attach itself to a ribosome. Now, a ribosome is a non-membrane bound organelle. It's made up of mostly ribosomal RNA, and what its job is to do is to take those codes of the mRNA and help produce, bring amino acids towards the ribosome. Now, the third step is called translation. So at this point now, we've got a strand of messenger mRNA, and it's bound to a ribosome. And the third step, this translation, is going to be when we get bits and small pieces of tRNA, transfer RNA, that have amino acids attached to it. And depending on the code of the DNA, a different tRNA will bring a different amino acid to the ribosome. In the ribosome, one amino acid is joined to the next one by forming a peptide bond, and then we move on to the next part of the DNA. We add another amino acid, another peptide bond, and slowly we start adding amino acids to this long chain. Eventually, this long chain of amino acids turns into a protein. So, just to recap, transcription is the first step. It happens in the nucleus, and that's where the code that's written on the DNA is transcribed onto a new molecule of mRNA, and then the mRNA leaves the nucleus. The next step, the next major step, is translation. So this is going to happen in the cytoplasm on a ribosome. The mRNA attaches itself to a ribosome, and then it moves along the long sequence of mRNA, and as it does, each three nucleotides, so adenine, thymine, cytosine, and guanine, every third one codes for one amino acid. So we move down three nucleotides, which we call a codon, and each codon will code for one amino acid. And we're going to talk a lot more about this in another unit later on in the course. Sorry. Okay, so here's a look at what happens for transcription. So I've talked about it, and let's have a look at it in pictures. So transcription takes place in the nucleus, so you can see the nucleus of the cell here. And we've got our DNA here, right? Double-stranded, double helix. The DNA will unzip, and when it unzips, we end up with two strands, 1, 2. The mRNA will form itself based on the ATGC code of the DNA until we form a new strand of mRNA. Now, when that mRNA is formed, it travels out of the nucleus and into the cytoplasm. So here's a close-up look. We get our DNA strand here. It's going to have the purple backbone. We've got our nucleotides here, the A, T, and G's and C's. So the DNA unzips right here, and we end up with our one strand here and the second strand here. So that exposes these nucleotides. So the mRNA comes along, and it uses its nucleotides to match up with the DNA. So it takes a code, it transcribes the code from the DNA, it produces this new molecule of mRNA, which will travel out of the nucleus and find a ribosome to attach to. So that's transcription. Once the mRNA, so here's our strand of mRNA, here's our strand of mRNA here. It's got the code from the from the DNA, it attaches itself to a ribosome, and in the ribosome, these molecules right here, which is the tRNA, they have amino acids attached to them. So as our ribosome moves along the strand of mRNA, they code for amino acids. So each time we see a different set of nucleotides, so for example, we could have AUG, we could have CUA. Each one of these three nucleotides on an mRNA strand is going to code for a different amino acid. So every time we see one of these amino acids right here being joined, it means there's three codons on that strand of mRNA. And once those amino acids join together, they form they start forming a polypeptide. So the little bonds in between the molecules in here, those are peptide bonds. And this keeps happening until we get a full sequence of amino acids, and according to the instructions on the DNA, that's going to be our protein. And that's the simple version of protein synthesis. So if you want to take one more look at it, we can have a look at this slide. Now this one's a little bit more complicated. It's got a lot more detail on it, but it's got the same principles. Alright, so you can see here, first of all, we've got the nucleus. So everything on this side over here is inside the nucleus. We get our DNA, and it unzips, and then as it unzips, we get the formation of a molecule of mRNA. And you can see how it matches up. So G goes with C, A goes with T, so on and so forth. When this strand of mRNA is produced, it's finished, it's taken all the instructions for that gene, it leaves the nucleus and it finds itself a ribosome. So here's our ribosome, and then of course this is going to be the close-up. So here is our strand of mRNA down here, and our ribosome moves along the mRNA, and as it does, our transfer tRNA, this green molecule right here, comes along and it has an amino acid attached to it. So this moves into the ribosome here. When it does, the ribosome forms this peptide bond, and as it does that, it adds on more and more and more and more amino acids. So here's our setup of amino acids down here, and then once we get to the end of the instructions, which are the code that was transcribed on the mRNA, that's when we end up with our completed protein. And that protein can do 100 different things, like I mentioned earlier. It could be structural, it could be an enzyme, it can be anything at all. So the last topic in Unit 2 is cellular reproduction. Okay, and this is how a cell reproduces, how it makes copies of itself. There's two main types of cell reproduction we're going to talk about. So the first is meiosis, and I'll just introduce this and give you the definition of this now. We'll talk about it a lot more when we get into the unit on reproduction. But meiosis is what we call sexual reproduction. So when meiosis occurs, and it only happens in certain cells in our bodies, the cells that produce our gametes. So in females, it's going to happen in the cells in the ovaries to produce eggs, and in males, it's going to happen in the cells in the testes to produce sperm. A meiosis is called a reduction division, and the reason why it is because the cells that are produced from meiosis, or cells that are produced through meiosis, they only have half the number of chromosomes as the rest of the cells in our body. And that way, when the egg and the sperm come together, they get half the material from the dad, half the material from the mom, and those chromosomes come together so they get a complete set of chromosomes. So that's meiosis, and we won't say anything much about that until we get to a later unit. Mitosis, this is asexual reproduction, and what asexual reproduction means is there's no combination or recombination of genetics. This just happens. The cells don't sexually reproduce. The cells produced are the same as the parent. Now, mitosis is used to produce somatic cells, and somatic cells are body cells. Those are the cells that make up our entire body. And in this type of division, the parent cell will split in half to give two genetically identical daughter cells. mitosis is a part of the cell cycle. And during the cell cycle, which mitosis only accounts for about 1 to 2% of the cell's life. The rest of the time, the cell is carrying out regular metabolic functions. It only reproduces, it only takes about 1 or 2% of its life while it's doing that. Now, to get ready for mitosis, however, it does completely replicate its DNA. So at some point during what we call interphase, which is the normal life cycle of the cell, the DNA gets completely reproduced. So we end up with double the amount of DNA. It turns itself down into, so we see interphase down here. We've got our chromatin. When the DNA reproduces, it coils itself up and it turns into the chromosomes. And the chromosomes are held together by the little tiny dot that you see there called the centromere. Now, there's 4 stages to mitosis. We have prophase, metaphase, anaphase, and telophase. During prophase, we get the coiling of the chromatin, and chromatin starts to turn into chromosomes. Or the DNA starts to turn into chromosomes. Also, we're going to have a disappearance of the nuclear membrane and the nucleolus. There's another, in animal cells, there's another organelle involved in this called the centriole. And a centriole helps organize separating the chromosomes. And centrioles usually occur in pairs. Now in prophase, one of the centrioles are going to move to opposite sides of the cell. Now, when we move to metaphase, the centrioles have connected to the chromosomes and they line them up across the center of the cell. It's a really easy phase to recognize because the chromosomes are well formed and they're usually lined up along the middle of the cell. During anaphase, we're going to have those chromosomes, the centromeres will split, and the identical chromosomes will move towards opposite sides of the cells. They're pulled apart by the centrioles. And then telophase is the last phase, or just near the last phase of mitosis. That's when the chromosomes move towards the centrioles. We'll start to have 2 new nuclear membranes forming around the 2 separate piles of chromosomes. The chromosomes will start to turn back into chromatin, and the cell membrane will start to pinch, and it'll form, eventually it'll look like a figure 8, and then once those membrane sides touch each other, they'll separate into 2 genetically identical cells. So let's just look at a picture of that to help you see. So we get interphase, and that's a cell usually undergoing normal life. Sometime during interphase, the DNA will double, so we'll have twice as much DNA, and by prophase the DNA will have turned into chromosomes. You can see the centrioles right here. They've moved to opposite sides. By the time we get to metaphase, the centrioles have organized the chromosomes so that they're right along the middle of the cell. By anaphase, those centrioles are going to pull back using spindle fibers, pull the chromosomes back to each side of the cell, and you can see the cell membrane starting to pinch off. In telophase, now we're having the chromosomes go back into chromatin. We're starting to see a nuclear membrane again, and then we get this pinch more pronounced. Eventually, this will pinch in the middle, and we'll end up with 2 cells that are genetically identical to each other. Now, one thing that we're going to do in this course is we're going to come back to some of the things that can go wrong. And very often, some of the things that can go wrong in our normal physiology is a disease called cancer. It's something we're probably all familiar with. We've probably all heard of somebody who's had it. And cancers are basically the result of cells that multiply really, really quickly. They don't follow a program that they should. Something happens, and they multiply really quickly, and as a result, they cause a lot of problems for our normal metabolism and our normal health. So here's like a bit of a background around cancer, and we'll come back to this theme throughout a lot of the different units in this course. And okay, that brings us to the end of Unit 2. Thanks a lot, guys.